cursor, the large one's myosin, the small dot's actin, and this gray shaded region between those two dots, that's where those cross bridges are forming. Okay, so we see a lot of things in two-dimensional, but it's actually more complex than that. All right. Now, another thing about that sarcomere is another tiny, well, we shouldn't say tiny, another protein called titin. Titin is sort of this spring-like protein in yellow that essentially acts as a recoil mechanism. When muscles contract, when those sarcomeres contract, they shorten. And so titin will allow that sarcomere to bounce back to its original length during the relaxation process. I can't extrapolate any more than that. Okay? Now, how are these muscle fibers being stimulated? So this is where we're going to have to rehash that neuromuscular junction again. But before we do that, it'll be important to know that muscle fibers, or a group of muscle fibers, are stimulated or innervated, is another synonym, by the axon terminals of one axon. Okay? So here is the axon of one neuron, here's the axon in another. Once they reach the end of the axons, they kind of splay out into individual axon terminals. Each axon terminal stimulates an individual muscle fiber. So all of the muscle fibers plus the axon that stimulates or innervates it is otherwise known as a motor unit. All right? So here are all those little axon terminals kind of diverging from that axon. Each one of these little dots is stimulating the muscle fiber. So this is a motor neuron, this is a motor neuron. Okay? Now, the number of motor neurons depends on what kind of muscle you are. If you're a rather large muscle, like your biceps, triceps, all your leg muscles, right? Those are, and the like, all of those are large muscles and hence have lots of motor units that will cause them to be contracted all at once, right? So having many motor units is going to be important for gross movement, not like icky. I'm talking about like very broad type of movements. Smaller muscles, like the muscles of your fingers, for example, have smaller motor units, which are important for the fine motor control or fine tuning of how you manipulate your hands and your fingers. All right? Fair enough? Okay. So this brings back to some nervous system nightmares, I'm sure. And so this is where the nervous system plays a particularly profound role in causing muscle contraction. And so that is the neuromuscular junction. All right, so at the end of that axon terminal, you've got this neuromuscular junction that consists of the presynaptic terminal, which is the axon itself. You've got this synaptic cleft, or this space, and then everything down here in yellow, that would be the postsynaptic terminal, or the muscle cell. All right, so what happens at this axon terminal? You've got this action potential coming down. And so we know that at this point, action potentials can't travel from one cell to another. Okay, you have to have those neurotransmitters that will help convey that signal from the neuron to the next cell. So at the muscle, skeletal muscle, acetylcholine, our favorite, or probably our least favorite at this point, neurotransmitter, is the neurotransmitter at that neuromuscular junction. It's stimulatory. okay? It's going to want that muscle to contract. Okay, fair enough? Okay. So this is something that I'm pretty sure I talked about, and if not, well, here it is anyway, all right? The book, you know, in all its flaws, does not do a very good job at kind of reminding the reader about this neuromuscular junction, at least in chapter 12. All right, and so here you've got this action potential, that orange arrow. It is flying down this axon, reaches one of many axon terminals. So what will it do first? It's going to start stimulating voltage-gated calcium channels. It has to be voltage-gated because an action potential, it's a change in voltage. So you've got the opening of these calcium, voltage-gated calcium channels. Which direction is calcium going to move? Into the cell. And we know, and we will continue to know, that calcium is a wonderful, very effective signaling molecule. And so it's going to tell those synaptic vesicles to start mobilizing, merging with the plasma membrane, and spilling out the contents, acetylcholine, into the synaptic cleft via exocytosis. All right, so you've got this acetylcholine that's kind of floating around in the synaptic cleft. It's going to have to do something. There, it will transmit that signal. It sits and binds to a sodium channel. What do we know about sodium channels? They open. Which direction does sodium flow in? In. Tricked you. Yeah. In. Okay. Into the cell, causing depolarization. Boom. Action potential on the other side. Okay. And then, of course, we know the rest of the story. Right. Acetylcholine kind of drifts away from that ion channel. It's been degraded by that nasty little enzyme, acetylcholinesterase. Choline gets recycled back into this presynaptic terminal to reform as acetylcholine. Okay? So if you've forgotten that, revisit that neuromuscular junction. All right? It's important because that leads to the mechanism of muscle contraction. Well, you need nervous stimulation. So there's this concept called excitation-contraction coupling. The excitation part was all of this. All right? And so I don't want you, I know I will probably confuse the hell out of you, but I want you to be able to discern which set of steps is occurring where, because they look very similar. All right? So we've generated this new action potential on the muscle side. So what? All right? So before we talk about the so what part, let's kind of revisit a little bit of skeletal muscle microanatomy, okay? So here is a muscle fiber. You've got your myofibrils, these tall columns in red, right? You have to remember that skeletal muscle is multinucleated, hence the blue nuclei, right? Since there's a collection of cells, there is a plasma membrane called the sarcolemma. There is a plasma membrane, cytoplasm called the sarcoplasm. All right, so a lot of these words are just going to have to repeat itself. Three other things that are important is that this sarcolemma, unlike your run-of-the-mill cell, will actually dive into the muscle fiber. All right, so here, this pale peach color, there's your sarcolemma. Here is where it will dive down into the muscle fiber. It transverses the width of that muscle fiber, and so those are called transverse or T-tubules. Okay? So when you have this action potential on the muscle side, it's traveling along the length of the muscle fiber, and some of it dives down deep into the muscle itself, muscle fiber itself. All right. The second thing are what's called the sarcoplasmic reticulum. And so it's a little bit different than your ordinary endoplasmic reticulum. This one holds calcium. Okay, so calcium is repeating itself over and over and again in the next 30 minutes. All right. So all that green stuff, that's the sarcoplasmic reticulum. Along that transverse tubule, you're going to find expanded parts of that sarcoplasmic reticulum. That's otherwise known as the terminal cisternae, okay, or cisternae. All right, so just kind of keep that in the back of your mind. All right, so we're done with the neuromuscular junction. We've got this brand new action potential that's propagating or moving down the length of the plasmic sarcolemma. It's going to also dive down into those T tubules. And so what will happen because of this depolarization? Well, just like the action potential on the neuron side, you're going to open up voltage-gated calcium channels. So calcium, not to beat a dead horse, moves in which direction? Into the cell. That calcium, that initial calcium that comes into the muscle cell, is going to stimulate the sarcoplasmic reticulum, here in yellow, stimulate the sarcoplasmic reticulum to release its own calcium into the sarcoplasm. Okay? All right. What else do you find in the sarcoplasm? The sarcomere. Okay? So now you've got lots of calcium floating around in this cytoplasm. And so what will happen? Calcium, whether it's coming in from the outside or from the sarcoplasmic reticulum itself, is going to bind to troponin. Okay? That will tell the tropomyosin to move a little bit, and then myosin can actually bind to the active site of actin, forming that cross bridge. Are you with me? Okay. Now, okay, so myosin and actin are interacting with each other. They're touching. That's fine. But a couple of other things happen. Remember, myosin has that enzyme ATPase. So now we have to think about our energy needs. So here at the top, calcium binds to troponin. The green blobby thing, that's your myosin head, ATP bound. I know it doesn't say ATP, all right? It says ADP plus inorganic phosphate. That's assuming that's the ATP molecule, all right? And so myosin will start to break down ATP, right? ATP, just for clarity, is broken down into ADP plus inorganic phosphate. And so here, your cross bridge, you still have ADP and inorganic phosphate, but for something else to happen, that phosphate has to leave, okay? When the phosphate is removed, you just have this myosin head that has ADP stuck to it. And so that is kind of like a signal itself. Myosin is going to shift its position a little bit. And what you get is something called a power stroke. Okay? Think of a power stroke as if you are playing, if you guys are divided into two groups and you're playing tug of war. Okay? Each one of you holding that piece of rope is a myosin head. And so what do you do when you play tug of war? You pull on it. That's your power stroke. Well, what else do? You let go of it briefly and you grab the rope again, pulling. Okay? And so that's essentially what actin and myosin are doing. They are literally kind of playing this tug of war game. And so once a power stroke has occurred, all right, it's yanking on that actin molecule, myosin is going to want to let go and reattach again at a different active site. But in order to do that, you're stuck with this ADP molecule. You have to regenerate ATP. So here, in this case, you've got the ADP molecule being switched out with a fully formed ATP molecule. And this cycle continues. You get this continual tug of war going on between these two proteins, provided that your signaling molecule is still attached to troponin. So you need calcium. Okay? You with me there so far?
How does ATP get reintroduced?
Well, your skeletal muscles have lots of mitochondria in it. All right, as long as you are undergoing aerobic respiration, you will continually produce ATP. Okay? All right, and so you continue to do this tug of war. So here is a sarcomere in its unrelaxed, unstimulated state. So here are your different components, the I band, the A band, the H zone. All right, so here's the I band with the Z disc on either side. Here's that A band. In the contracted state, because you're playing tug of war, you are increasing the overlap between these two proteins. And so what happens? The sarcomere shortens. So look at the I band here. The I band is smaller in width than in the uncontracted muscle. Okay? The sarcomere shortens, but what's important to realize is that the actin and the myosin do not change in length. Ever. Well, maybe not ever, but during muscle contraction. So think about tug of war again. If you're pulling on this rope, is the rope changing in length? No. Okay? So neither one will change in length. You're just increasing the overlap and thus making that sarcomere a little bit shorter. Okay? Yes? No? Next week? Okay? All right. And so it's important to kind of understand this setup. This is a story. Start from once upon a time there was an action potential. All right? Then you get muscle contraction, the end. And if you're able to, if that means you're going to revisit that neuromuscular junction, hammer it out separately before talking about muscle contraction itself, that's fine. But you got to be able to understand that sequence of events that takes place from the nervous system down to the muscle cell. Okay? So, that, in other words, star this slide. And the things that were before it.
The sarcomere is the only part that's shortening, or is it just overlapping?
The sarcomeres don't overlap.
It's just the I band.
The
The
I
actin-
band,
The actin and myosin, I mean.
yeah, the actin and the myosin will overlap.
But the sarcomere is short.
And that way the sarcomere shortens, but the length of those two proteins doesn't change.
They just overlap on top of each other.
Yes. Okay? Okay. Well, how do you relax? Well, you got to get rid of your signaling molecule. All right, so calcium has to detach from troponin. All right, that tells tropomyosin to shift back to its original position and cover those active sites. And so calcium is still hanging around, and so it'll either go back to the sarcoplasmic reticulum through active transport, all right, so you need energy for that, or it'll leave the muscle fiber, the muscle cell itself, via calcium, sodium transport in the opposite direction, okay? All right, if you don't remember anything else, troponin, calcium detaches from troponin, boom, no more muscle contraction. The signal's gone. All right. Okay. All right. Whew. That's a lot of signaling steps. So let's shift gears a little bit and kind of talk about the different types of muscle responses. Let's start with the easiest one. The simplest, I shouldn't say easiest, but the simplest muscle response is what's called a muscle twitch. All right? If you have a stimulus that is strong enough to cause an action potential, then a muscle fiber is going to be stimulated. Okay? That's basically what a muscle twitch is. And a muscle twitch has 3 sections. It almost kind of looks like an action potential itself, sort of. All right? The first one is what's called the lag or latent phase. This is a time that occurs after a stimulus has been applied. So here, time versus tension. This little arrow here, that's your stimulus. Okay? This lag time is the time that it takes for calcium to kind of mobilize and present itself as a signaling molecule and bind to troponin. Once that happens, boom, you have an increase in muscle tension. That's the contraction phase. Okay? After a while, it kind of slopes down. That's relaxation. That's the time that calcium is removed from the troponin complex. Fair enough? Okay. So muscle twitches, the simplest type of muscle contraction. There's another thing, here, this one is from a previous lecture that I gave. This one is actually from your book. So the same thing. You've got a stimulus, you've got a short lag time, and then you've got your muscle twitch, okay? There's another type of muscle response, which typically occurs when a muscle has been kind of in a relaxed state for quite some time. And that's called TREP. TREP, I think, is derived from some French word that means staircase. And so what happens is, in TREP, you receive multiple stimuli of the same strength, okay? And so the first muscle twitch is going to be of some arbitrary tension, but undergoes a relaxation phase. But because it gets stimulated, that muscle fiber gets stimulated again right after it, the next muscle twitch is going to be a little bit higher in tension. And it continues to increase as another stimulus is applied until you've reached a constant tension, whatever that value is. All right? So a TREP is like a staircase. It increases after so many stimuli. It'll increase the tension in a stepwise fashion. All right? Okay. Now, there's another type of muscle response that's kind of like a TREP, but not really, and that's what's called summation. Okay? If you look back at TREP here, you have a lag phase, you've got the contraction phase, you have a relaxation phase. And you go through that same cycle each time in TREP. In summation, okay, your muscle hasn't had enough time to relax, okay? In other words, not all the calcium has been removed from the system. So if you get a stimulus here that results in a muscle twitch, it starts to undergo the relaxation phase but hasn't quite finished. Then you get another stimulus. Then the second muscle twitch kind of piggybacks off the first. So you get an increase in tension. Okay?
Is it kind of like a cramp?
It's going to be. in the next slide, all right? Okay? So that is what's called summation, right? The muscle hasn't had time to relax. Now, that leads to something, if you are increasing the frequency of those action potentials, now you're going to experience something called tetanus. Don't be confused by the disease, okay? The disease causes constant muscle contraction, but it's rather painful and doesn't stop, okay? This kind of tetanus is rather short-lived in relative terms, all right? So if I were to apply some sort of stimulus to muscle fiber, I'll start out rather slow, 5 shocks or 5 stimuli per second. I'll have a number of twitches, okay, in succession, all right? This kind of muscle contraction is what's otherwise known as an incomplete tetanus, all right? So it's kind of like summation, but you have those individual twitches. It doesn't completely relax. So you get this increase in tension. It's almost kind of like jerky movement in terms of muscle contraction. If I increase the frequency of that stimulus or stimuli, then I'm going to increase the frequency of those successive muscle twitches. So the frequency here of those muscle twitches increases to a point where you can't even discern those muscle twitches, okay? But if I now apply even a greater amount of stimuli, all right, then I can undergo what's complete tetanus, where you don't see those peaks of muscle twitches because the cell or the muscle fiber never has an opportunity to undergo the relaxation phase. So the muscle in this case is constantly contracted. It has reached a maximal tension and it stays contracted until you reach a point where it gets tired. And so you get this decrease, jagged decrease in tension, and that signifies fatigue, all right? By this time, yes, you would probably have muscle contraction. So the disease tetanus, the disease that causes tetanus, is very similar to this, all right, except that you don't have the fatigue phase. It's constant, hence it's painful, all right? And that's caused by something that happens at that neuromuscular junction, okay? All right. Are there any questions? Okay.
So the complete tetanus would be the example of cramping, because you're contracting for so long.
Mhm.
Basically just
You don't have time to relax and you get tired, okay? You got that buildup of lactic acid. You're not utilizing aerobic respiration effectively, and so your skeletal muscles are starving for oxygen. Okay? All right. So I gave you a lot. This is where I left off in the other 2 sections. So it's Friday, our Friday at 4:45. It's crappy outside, so go hide underneath the blanket and stay warm. All right?